Beyond polygenic risk, why genomics may need a broader model of causality. Human genetics has become extraordinarily good at finding associated variants, but it has been less successful at answering a harder question: what kind of biological system produces those associations in the first place? For years, much of the field has worked inside an implicit model. Complex disease is often treated as the sum of variant effects, where the most statistically prominent loci are interpreted as the most important. At NomosLogic, we have been studying a different possibility: that systems are organized not around a handful of variants, but around distributed constraint architectures, interacting sets that preserve stability even under perturbation. That distinction matters because it changes the question. Instead of asking which variant is most prominent, it asks what kind of system can preserve stable behavior under perturbation. Using PROTEUS, our deterministic evolutionary simulation framework, we tested whether genomic systems collapse under targeted perturbation or reorganize while preserving convergence. The results were striking. Across the observed dataset, 88.5% of systems exhibited distributed architecture. In cardiovascular tests, removing the dominant contributor preserved 99.996% of peak fitness. Across perturbation experiments, no exclusion caused loss of convergence. In fact, the exclusion produced 29 novel variants rising into top-ranked positions while accelerating convergence from generation 140 to 90. Validation against empirical null distributions yielded an AUC of 0.9386, indicating that the framework was identifying biological signal rather than optimization artifact. That pattern is difficult to reconcile with a simple variant-centric model. It is much easier to reconcile with the idea that stable biological behavior emerges from distributed interacting architectures. This is also why determinism matters. Many computational approaches in genomics are stochastic. If identical inputs do not reliably produce identical outputs, then it becomes harder to distinguish biological structure from computational noise. PROTEUS was designed to be deterministic. The goal was not simply to optimize a score, it was to test whether convergence is an intrinsic property of the system itself. The evidence suggests that it is. If this view is right, the consequences extend beyond theory. Variant interpretation, therapeutic targeting, and risk modeling all depend on how causality is framed. If the field overweights individual contributors, it may target concentration points rather than structural requirements. A target may be associated, yet still not be the right abstraction. Genomics may now need to ask a different question. Instead of asking which variant is most important, ask what kind of system preserves stable behavior, even when its most visible contributors are removed. The work ahead will be about identifying how biological systems remain coherent, how they reorganize under pressure, and how that organization can be measured without confusing prominence for necessity. Genomics does not need less statistical power. It may need a broader model of causality. In many genomic systems, causality may be distributed across interacting sets rather than concentrated in a few dominant anchors. And move beyond ranking logic toward architectural logic. Visit nomoslogic.com to learn more. NomosLogic, deterministic, exact, yours.